My name is Richard Seabreeze, and today we'll be talking about atomic force microscopes, or AFM. So AFM allows you to image the surface of samples. You can zoom in much closer than you can optically, so you can go beyond the optical limit of light. So the AFM is able to produce an image of our sample surface, or the height of the atoms on the surface of our sample, by feeling the interacting forces between a very sharp probe and the actual atoms on the surface of our sample. And that occurs in this little region here. Here's an as seen image looking through the AFM optic. Here we have our sharp probe tip, and it is hovering over top of a fungus sample. My current research is partnering with a group from UMBC, looking at mycelium materials and their physical properties associated with them. Here's the actual AFM data of one of the fungus samples. So this is a 50 micron by 50 micron scan of one of the fungus samples. It is able to create a false color image using lighter regions as higher, corresponding to higher, and darker to lower, to plot out the height of the surface. So here's a schematic showing the growth process to make samples to look at with the AFM for this filamentous fungi. So starting up at the top, we thaw out our samples from a spore bank, we grow them in liquid solution, and eventually we plate them for imaging using the AFM. So here's a poster that I presented on the work that was done in this fungus project up to this point. It was presented at a annual physics conference, the March meeting this year in 2024. It was held in Minneapolis, Minnesota. And again, this recaps all the work done up to this point and the results. This is me presenting the poster at the conference in Minneapolis, Minnesota. It was a very large conference, very good time. My first time presenting in this type of format. Um, Many people walked by, had a lot of questions, was able to speak with a lot of folk and get some good insight into the project. Here's myself and all of the Towson group that traveled to the physics conference in March to Minneapolis. We all had a great time. Everybody presented, gave talks on their individual works and their contributions to the field, and it was a great time all in all. So this is where my journey in AFM started, at the old building at the Towson campus. This is the Smith Hall physics level, and this is where the AFM originally sat that I worked on. You can see some scribblings in the background there and some other instruments lying about. Here's the new AFM station currently set up in the new science complex at the Towson campus. This is where the majority of my work's been done. There's 3 instruments in the AFM lab, 3 AFMs, but this is the current one that I've used, a Bruker instrument, and it's got the capability of taking large area scans. Here's one of the newer instruments recently purchased with the new addition of the science building. So here is a Cypher Asylum AFM, and this has environmental control capability, and it has this acoustic isolation that allows you to take atomic resolution images very easily. So third, last but not least, this is the Bio MFP atomic force microscope. It has the capability of imaging petri dish size samples. It also has, in conjunction with the AFM, an inverted Zeiss fluorescence microscope. So getting back into the images of some AFM data here, currently we're looking at height images. So again, false color, the lighter regions correspond to higher areas of the sample, darker to lower. So this is the top-down view of cancer cells, 2 cancer cells together there. That's a project that we're working with a group out of Rockville. This image is a zoomed in image of the previous photo. So now we're looking at about a 40 micron scan of the what is dried out cancer cell. So this is a breast cancer cell that we got provided by a group out of Rockville, Maryland, to do some collaboration. Again, some more height data. So again, false color height map representation of, in this case, this is small polymers on a hard substrate surface. So you can see the small polymer spheres deposited on this flat surface substrate. The interesting capability of my instrument, my AFM that I do work with, it has the ability to do force mapping at all points on the height data image. So now this is the same sample. Instead of height information, we're looking at deformation or other, how far into the sample are we pressing with our tip at each point on the image. Looking at some more samples, now this is the optical image. So looking at our setup, we have our, in the black, um, this tip shaped structure, that's our tip, and that's hovering over top of a very flat substrate, some crystals growing on a substrate. So the previous image was the optical setup of this sample that here that we're currently looking at. The old image was about the scale of 1,000 microns. This here, the scale bar is on the height, the size of the scan here is 1 micron by 1 micron, and that's the height data of that crystal. So here is a different crystal structure. Scale bar again at the bottom of the screen showing that that represents 600 nanometers. So we're looking again at the height, otherwise known as the topography of a crystalline structure grown on a substrate here by a group at Towson in the physics department. Here's an AFM image of a DNA strand, so it's showing the actual double helical structure found within DNA. And again, AFM, atomic force microscope, so it's imaging the interaction between the atoms, and it's able to produce this false height image using false colors.